scholars from SB and other colleges. I also thank the organizing committee, especially its secretary, Dr. Philip Litto Thomas, for organizing this program. Wishing you all a fruitful hour. Thank you. Thank you all. Over to Josie.
Thank you, Jomon sir. I would like to invite Dr. Philip Litto Thomas, Assistant Professor, St. Bergsminks College, and the organizing secretary of the webinar, to introduce our guest speaker of the day. Sir.
Thank you, Josie. Teachers and dear students. Today we have a resource person from the University of Cambridge with his very roots in Changanaശ്ശേരി. Dr. Joseph belongs to the Thottacherry family of Changanaശ്ശേരി, and interestingly, his ancestral house is very near to a college at Madhumula. Dr. Joseph did his master's in biotechnology from VIT University, Vellore, after which he spent a short stint at Dr. Pradeep's lab at Rajiv Gandhi Center for Biotechnology, where we got acquainted with each other. Later, he moved on to NCBS Bangalore, where he did his PhD with Dr. Satyajit Meyer, the then director of NCBS. And his doctoral work focused on endocytic pathways of cellular membrane. After his PhD, Dr. Joseph joined the laboratory of Daniel Johnston at Gordon Institute at the University of Cambridge for his postdoctoral work. And currently, he is engaged in studying epithelial cell dynamics using novel tools that can shed more light on many malignancies, including cancer. And Dr. Joseph has many high-impact publications to his credit, including those in Nature Communications, PLOS ONE, etc. And he's a recipient of many research awards and travel grants from India, Europe, and the USA. And we are very happy to have this young scientist in our midst. And I hope that he can be a motivation to the young minds listening to him today. Wishing everyone a very fruitful session. Thank you. Over to you, Dr. Joseph.
Hello. Can you hear me?
Yes, yeah, you're audible.
Yes, let me just. Can you see my screen?
Yes, yes.
Okay, great. All right, hello. As you heard, my name is Joseph Thottacherry. I'm a research associate at the Gurdon Institute in the University of Cambridge. It's a great pleasure to be here talking to you guys. Unfortunately, it's over Zoom and not in person, but it's really a great pleasure, and thanks to the organizers, especially Philip Litto, for inviting me over here. Today, I'll be talking to you about how we watch Epithelia in action. And I understand that you guys are not at college and are at home and watching from your tablets or phones, and I'll try to keep it short. And if you have any questions, you can ask me later after that. So why epithelia, right? Epithelial cells, as you might know, these cells form sheets and line our organs. It helps separate and protect the inside from outside, but also works as an interface. For example, in the lungs, it lines the lungs, and it helps in exchange of gases, or in intestines, where it helps break down food and absorb nutrients, or as the outer layer of your skin in sensory perception. So these epithelial cells are held very tight and acts as a barrier against microbes, if you will. For example, on your skin, when you're touching somewhere, or in your lungs when you're breathing, or in your intestine when you're eating something, right? So it's held very tight and forms a barrier. But these barriers are under constant attack. They're always being invaded by bacteria and other pathogens or toxins and being killed regularly. And this needs to be renewed efficiently so that this breach can be renewed, right? So how do these epithelia get renewed? These epithelia cells are usually renewed by stem cells. So stem cells shown here is like is an undifferentiated cell that has potential to become many different cell types. So I'm showing you an intestinal epithelium here as an example, and here what is marked is an intestinal stem cell that is written here, and it forms these enterocytes, which are the major cell type of this epithelium, and that it forms by forming an enteroblast. So the intestinal stem cell differentiates into an enteroblast, and then it forms into an enterocyte. And this balance, so if an enterocyte is lost, it needs to be replenished back. And this balance is very fine and needs to be maintained very carefully, or there can be diseases. For example, if instead of one, it makes multiple cells, it can form hyperplasia, where there are more number of cells, which can cause a tumor, for example, in like colon cancer. Or you can imagine that these cells do not form these very tight junctions, or septate junctions as it's called in Drosophila, and that can cause leak in the gut. So things could just go in, or microbes could go in, and this can cause a lot of diseases. This is nicely shown here in this mutant and wild type Drosophila case. So this is a wild type Drosophila, and this is a mutant one. They're both fed with this blue food. So they're both eaten this blue food. You can see that the wild type, you do not really see the food anywhere. It's in its intestine. But in this one, you can see that it's being leaked all over its body and all over. And so this is called a Smurf assay in relation to the Smurfs. So then the question really is, how does this polarized enterocyte form, or polarized epithelial cell form, right? So there is this unpolarized tiny stem cell, which then forms into these enterocytes in a very precise manner. And the first question is, can we see it, right? Can we see these cells forming this, so that then we can start to understand how these processes happen? So in the sense, in a high temporal resolution, we need to see it live, happening live in real time. The second thing that we would want to do is watch these changes in high spatial resolution. So here is an enteroblast, right? Enteroblast that doesn't have these sort of junctions with these. So which means that this cell, when it becomes this one, it needs to somehow start making these. And it needs to invade the space between these two enterocytes and go in and form this enterocyte-like structure by forming these septate junctions. So we need to see it in very high spatial resolution as well. And for these, I used Drosophila, like I showed in the previous slide with the Smurf assay, to understand these processes. We used the Drosophila intestine, specifically the midgut of it, to look at that. So you might be wondering, why Drosophila? So Drosophila, or the fly, as it's generally called, is a powerful model system. It's very tiny. You might have seen it on fruits, like in your home, especially during summer. It invades all your fruits. But why we are interested in it is because it is very similar to us. The fly system is very similar to us. It is very similar in terms of having similar organs, or it has very similar genes between us and them. So you can see that the digestive tract here is very similar in fly and humans. The nervous system is very similar in fly and humans, and it's used regularly to look at how nervous system is organized, how muscles are organized, or even how the body plan is organized. Like when you go from an egg and to a full animal, how is a single cell organize itself in an anterior posterior direction and form a head in this side, and the leg in this side, and the wing in exactly here. So how is this body plan organized? Why do we form 2 legs and 2 hands, and why is the head there? So these things are nicely studied using a fly. So it's a very good system, and it's tiny. You can see with housefly. In the lab, we grow these flies in these vials. You can grow thousands of them very easily. You can just keep it in this vial with a cotton plug and just leave it at 25 degrees. And in room temperature, really, just leave it in the lab. And the interesting thing is that its life cycle is so small. So in 25 degrees, in 10 days, it goes from egg all the way up to adult. Sorry? Okay. So if you're interested in, say, looking at a gene that's causing a disease in humans, you can't obviously study it in humans. So what you can do is go take a Drosophila, make a mutation in that gene, or make a transgenic line or tag it, and then study what happens to that gene in that particular process. So you can then ask, how does muscle form, or why is this nervous disorder happening, right? So this is very useful. And in our case, it is quite interesting because the Drosophila intestine is very similar to a mammalian intestine. So we could make parallels between them and study this very easily. The second thing, as I said, is it is extremely genetically tractable. You will see it in later slides as I show you that we can tag it, we can make mutants and things like that. So this is a very good system, and this is where we do our experiments. So, I mean, as you know, how do we do experiments? It's the foundation for all discoveries, and it's very simple. You make an observation, you have an idea, and then you test it, right? You perform an experiment and you see, oh, does it work? And then you get something and you tell others, oh, this is what I found. Can you actually see these things? So this is what we do. And this is just to keep in mind that this is the process that we continuously go through. So coming back to it, the questions, I'll first focus on how we image them live. And second, on the high spatial resolution ways that we can observe this. So can we image them live? So there are times where you can ask a question in science and then just go ahead and answer it. You want to ask if a protein gets phosphorylated. You can just do a Western blot or use an antibody. But there are situations where you need to start creating tools or start making ways in which to address a question. So this is what we found, that there is no real way to image them live, and we need to develop a new method to actually image them live. So this is what I tried to image them live. And I quickly realized that the intestine, if you want to visualize the intestine, the cuticle of the fly is too thick and you cannot really image through that. So I dissected the cuticle out and exposed the gut in like this, and embedded them in agarose and in cover slips so that it is immobilized. So there's a cover slip underneath it and there is a cover slip over it, and you can image it from below using a fluorescence microscope. And these can be kept in these 4-well chambers, which is very tiny small, and you can then image like multiple flies at the same time. Now the next challenge was, how do you maintain the fluid composition here, right? You need to do test different things so that it is very similar to the body composition over here, or the hemolymph composition over here. So we made an artificial hemolymph which keeps the fly alive. And we were quite happy to see that the dissected fly was keeping alive for many, many days in culture. Here is a dissected fly which is happily munching on yeast and is completely fine. So we could now use this as a system to understand how the gut renews, right? So this is the fly, and you could just go into with a microscope and look at that. This is just a bright field image, but what you can immediately see is these beautiful peristaltic movements in these gut. So peristalsis is where the muscle cells around the gut is compressing it so that it can push the food down. So from the mouth down, it can push the food so that it gets broken down by the cells and it can absorb the nutrients. So peristalsis is very important for that. But here it is showing us that our system is working very well. But what we're interested in is really the cells over here, right? We are interested in these cells and then understanding how the stem cells here can become endoblast and become endocyte. So for that, we need to start seeing the cells. And that's what we did. We can now visualize each cell, which is seen here in red. Each is each of the nucleus. But wait, how do we do that, right? We use something called fluorescence microscopy. I'm sure you know about it, but just let's go through it for a second. Before that, how do we even see a color, right? When you see a red ball, what really is happening is that the white light is forming, which consists of all these different wavelengths, from blue to green, blue to red, from the low wavelengths to high wavelengths. And this red ball is essentially absorbing all the wavelengths except for the red, and it's reflecting the red. So then you see it as red ball. So that's what's happening. And when it is a white ball, what is happening is it's reflecting all the wavelengths and from blue to red, and that's what is happening. And the black is really not a color, it's just absorbing every wavelength and you're not really seeing anything. So lack of seeing something is black. But fluorescence is slightly more interesting. It's where a molecule absorbs in a particular wavelength. So instead of all these different wavelengths getting absorbed, it's absorbing in a particular wavelength, here let's say green, and it emits in a higher wavelength, in the red wavelength. So this is lower wavelength, this is higher wavelength, even more higher would be like infrared. So that's what it's doing. So now what you can do is use a microscope and send in this light, green light, and then visualize it in red, right? So that's what we do. You send using a laser, you send in a green light, and use an objective to send it to your specimen, which is your gut in this case, and the fluorophore, which is here, which will emit in the higher wavelength, you just capture it using a camera. And that is it. You can now visualize it, right? So you can go back to this image that we saw before. So what you can do now is express these red fluorescent proteins, which is a fluorophore, in these nucleus. And for that, what you do is you just attach it to a protein of interest. For example, here, what we're doing is histone. So these protein, histone, is tagged to red fluorescent protein, and you can express it in the entire fly, and all the nucleus in the fly will be labeled. And this is amazing. You can now see each of the cells, and you can see it is doing peristalsis very nicely. But our question is really to see these small cells becoming big, but here you can't really see the small cells. You can see the big cells very well, so you need to zoom in, right? But before I go into that, I just want to tell you a bit more about microscopy in case some people are interested. So what we saw before is where you're sending in a light, a lower wavelength. It is going in. There is a fluorophore here, which is emitting in a higher wavelength, and you're collecting it. But as you can see, it's happening in one point. And now if you want to see the entire gut, you need to go one point, next point, third point, and so on and so forth until you can image the entire gut. But this is really slow, isn't it? It's like how you get a paper printed out, first line comes printed out, second line comes printed out, third line comes printed out, which is very slow. Imagine you're watching a movie like this, where only the top line comes, then the second line comes, and so on and so forth until you fill the screen. Then the next frame happens. Really small, it's really slow, right? So we use something called spinning disk fluorescence microscopy, where instead of sending in one light at a time, one point at a time, you're scanning multiple points simultaneously, and that we use this disk. And interestingly, this was initially used in TVs to create images. And we have a third technique, which we use quite often, where instead of sending light through an objective and collect it back through the objective, what we do is send the light perpendicular to it. So you're essentially illuminating the entire field that you're interested in using one objective, and you collect it using a second objective of this entire thing. So which means that you just shine it and the entire thing is collected, so which is quite fast. You can just keep that in mind, and if you have other questions, you can ask me later. So that's what we do. I use spinning disk to do these images. And so going back to our initial problem, we want to look at these small cells, not just the big cells, and watch it becoming into the big cells, right? So I zoomed and used a higher resolution, and now you can start seeing that. So you can see the big cells, the nucleus, and you can also see the small ones, right? This is a small one that you see. But it's really moving a lot because of the peristaltic movements. The gut is moving quite a lot. And if we want to actually visualize this and track it forming into another cell, this is really impossible here. So we need to somehow stop the peristalsis. We need to stop the bowel movement. And we know that the fly is very similar to humans, right? So could we then use something that we use in humans to stop the bowel movements in fly itself, and try that? So we use loperamide, which you would know as loperamide in India, Imodium here, to stop the bowel movements, and we use that in the fly and fed the fly. And what you can see is that it actually works. Here you can see the time is going forward, but the gut itself is quite still. And you can see the small cells, which is here, and the big cells, which is there. And now you can imagine tracking them and watching this form into this. But how long do we image for? Do we image for minutes? Do we image for days? Do we image for weeks? we don't really want to image for a long time, right? We want to get it really fast. So how do we do that? How do we reduce the time that we image, but we can see more number of these stem cells forming into enterocytes? So we found that the gut, when the Drosophila is born, or a newborn Drosophila, you can see that the gut is really small. But in even just 2 days, it forms into a really long tube, which means that a lot of stem cells are actually becoming into enterocytes during these 2 days. So which means we have higher chance of finding this event happening. So we could then look at a stem cell forming into an enterocyte during this transition. So how do we look at stem cells? So we need some way to just look at stem cells and then track it over time, right? It is right now sitting like a cyclist like this, which is in the dark. You need to somehow bring light to it. And this is another situation where Drosophila is a very good system, and you can modify it in using a flip-based system, details I will not go to right now, where you can just express a fluorescent protein only in these cells. So what the trick that you can do is you keep the flies in 18 degree in an incubator, and there is nothing expressing, but you can express green fluorescent protein only in these precursor cells on command, and you just shift it to 29 degree for 2 hours, and it starts expressing this. And then all you need to do is watch these cells over time. It will form something like these big cells. So that's what we did. And here you are seeing the anterior side of a gut, okay? Anterior side of a gut. And here you have expressed both these. You have expressed GFP in these precursor cells like we saw before. But at the same time, we have expressed red fluorescent protein in all the nucleus like we saw. So you're basically combining the two things that we saw before into one fly. And so then you can start imaging, you can see the cells at the same time here. So you can see all these small cells in green are these precursor cells, and the big ones you can see are the enterocytes. So this is the day one of you have shifted it. So this is literally day one what has happened, and I'm just going to show you a Z section of the gut, so you can see through the diameter of it. So this is the bottom, and now it has come to the top. So this is the diameter of the gut, and this is the length of the gut, and you can see these green cells, which are these ones, and the big ones. So this is forming the side of the tube. But if you now look at these cells in the next day, what you see is that these cells have become into these big cells suddenly. So in just 1 day's time, it has become into an enterocyte, which means that all you need to do now is just watch it continuously for the entire day. So you can see it slightly more better in the posterior side of the fly gut, where you have way more stem cells, and you can see again these stem cells, and you can see the big enterocytes. But in the next day, if you watch these in the next day, you can see that a lot of stem cells have become into enterocytes. So which is fantastic, which means that now we can image them live with a high temporal resolution. You can image them for days and then watch and understand how stem cell becomes into an enterocyte. Going on to the second part, so can we see the intracellular changes, right? Can we see see these in high spatial resolution? This becomes quite important because what we see during this transition is that these enteroblasts, when they become into enterocytes, they actually form this apical site inside it. So it has preformed the apical site inside the cell, and all it does when it becomes into big one is just expose it over there. But how does it happen? I mean, how does these junctions, which is actually forming the barrier, remodel? How does this one break so that it can just form? But then, it needs to form between these two ones immediately so that there is no leak that is happening, right? So this is very important. So we are not interested in the bottom part of the cell. We are really interested in the top part of the cell or the apical side of the cell. But unfortunately, using the confocal microscopy or all the usual standard fancy microscopy that you use, the top side of the cell is really blurry. So you can see a similar section here taken by Mihoko, where you can see these enteroblasts which is forming, with this apical surface, and it is forming these ones. And you can see there is some some fussy thing happening here, but you can't really see it. So can we really see it in high resolution over here? And this is an inherent problem with microscopy. So this is a bead, a normal bead, which is 200 nanometer. It's a normal fluorescent bead and 200 nanometer diameter. You can see that you can see it nicely in XY. But as you image in Z, or you go in depth, you see that the resolution is really bad. It cannot image it properly. So which is a problem, because what we want to do is essentially image it in Z, right? We're not interested in XY. We want to go up and see what's happening here and in the gut. But to just give you a visual perception of this, the gut is actually going to be like this, right? You're going to cut the gut in longitudinal, and you're going to see it like this, and it is this portion that you're interested. And here, what happens to the septate junctions, or the junctions, and the enteroblast is what you're interested in. So how do we solve this issue? So we decided to use something called expansion microscopy. So expansion microscopy is where you embed your sample in a gel. So this is that gel before. And what in essence, what you do is it's a water absorbent gel, and you put it in water and it absorbs the water and expands. And when the gel expands, because you're cross-linked it with your cells or tissue of interest, the tissue also will expand. This is something you might have seen in the roadsides in Thottacherry, where people sell these tiny beads, which if you put in water becomes bigger, right? So this is essentially what's happening here using sodium acrylate, which is water absorbent. But instead, when it is in this stage, you put your cells inside that and you just expand it so that you can see it bigger. So imagine you have Golgi and mitochondria, which are inside the cell, which are tiny. You can expand it and make it bigger. So we wanted to do that for the gut. So can we expand? So this is done in a cell culture. Nobody has done it in the gut, in the tissue. So we wanted to standardize that protocol and make it work here. So can we expand the gut and then look at this apical side and ask what happens to the junctions and these ones? So this is in collaboration with a collaborator in the Yale University, and this is the protocol that was published, where you can go from this size and you can embed it in the gel and then just expand it. So you can see the size difference that you get over here. So can we do that, right? So this is the gut, which was expanded once, and then this is a second expansion. So this is all the nucleus that is labeled over here, and then it is expanded, and you can see that it's slightly expanded. But what we really want to do is reorient it so that the apical side becomes the basal side. So we want to, so you have the gut, you cut it, and this is cut, and then you flip this 90 degree. So what will happen is that you have cut through the diameter of the gut and you have flipped it. So whatever was on the top side, the apical side, will now become the basal side. And now you can image it nicely. So this is actually nucleus, which is labeled. So each thing, so this is a nucleus, this is a nucleus, this is a nucleus, this is a nucleus in before expansion. And after expansion, that single nucleus becomes of this size, which means that you don't need a fancy microscope to actually see it in higher resolution. Now then the question is, what about this image, right? So what happens to these apical sides and how better can you see it? So what we can see is that it is in really high resolution, and these blurry things that you see, you can see it as these nice microvilli over here, which means that we can now image them very well and see these in high resolution. So we could go back now and then ask how these cells form into these enterocytes and how we do that. So this we can now do it in high temporal resolution. Watch, how fast it becomes this. How does it push these cells? How does it become that? Or we can do a high spatial resolution where you can just look at a cell and then ask how it goes into that. So this is what we can do. So we are very successful in developing these methods so that we can answer these questions. And I hope to talk to you about what we find later on. But so what we plan to do now is, as I said, we can now look at each of the cells, but we can also label each of the cells separately. So we can mark stem cells, enteroblasts, and enterocytes separately here. by using 3 colors. So we can have red fluorescent protein, which marks enterocyte, and we can have stem cells where it expresses both red fluorescent protein and cyan fluorescent protein, or we can have endoblast, which expresses all 3 combinations. Now, by looking at which combinations expressed in which, you can label them as either here stem cells, red stem cells, or the yellow is enteroblast, and the gray is enterocytes. So which is very nice. So you can now just watch one cell become into other using the new system that I have developed, and then ask how this cells are formed. So that's all I wanted to tell you today. So thanks to the funders and Daniel for the mentorship, and this is Gurdon Institute at the University of Cambridge. I'm sure it might be quite difficult to follow through Zoom and through your mobile phone. So if you have any doubts, you can write to me either on Twitter or on email, and I'll be happy to help you. And thank you very much. This is it.
Thank you very much, sir. Sir?
Can we go for some quick questions, right?
Okay, yes, sir. Participants can either type their questions in the chat box, or they can unmute their mic and speak up.
Yeah, Joseph, I have a very basic question.
Yes.
You dissected the flies, and they still live for 5 days, right? And
yes.
how do they live? Their nervous system also got damaged, right? And how does it happen?
So we make sure that we don't damage any of the organ systems. So we just remove the outer cuticle. So this is the trick. So we need to do the dissection using a very fine method so that you don't damage any of the organs. So you can see that even slight disruptions can affect your gut itself, and the gut can just burst open. So this is a very fine thing that you need to do. Yes.
Okay. Yeah. So you just remove the cuticle.
Yes.
And extend the digestive system.
Not so much. It is just for visual purpose. I've shown you that way. We won't extend it so much because there is trachea that comes into the gut over here, and it is very important. So in fact, we know that the trachea and the oxygen transport is important for the stem cell divisions and things like that. So we don't really extend it so much. There is also Malpighian tubules which goes around, but then you will not be able to understand what I'm talking about. This is really for visual purposes that I have removed them to show you that here.
Okay. Someone else has a question? Amal Samuel, would like to have a question?
I think there's a question in the chat by Anoop. We can image them live. Is it because of the fact that Drosophila is immobile during its embryogenesis? Okay, that's a good question. What I think I didn't make really clear is that this is not embryogenesis. What we are seeing is an adult Drosophila. It's after it's been born, and it's not in pupae state, any other state. So it's an adult Drosophila that we are watching the gut in. So this is why we can look at it. Yeah.
Excuse me, sir. Hello, sir.
Hello.
Can you hear me?
Yes, I can hear you.
So actually, you said like you have tagged histone proteins for visualizing the cells, right?
Yes.
So like, it's not only in the epithelial cells that has the nucleus. So the rest of the cells in the gut has also nucleus, right? So
yes, in fact, the entire all the cells in the fly will be labeled, not just in the gut. Yes, that is correct.
Okay, so here you visualize the cells, not only the epithelial cells, rather than the other cells also you have fluorescently labeled, right?
Yes, correct. Yes, but we are interested in seeing only this, and so we are just looking at it. But yes, every other cell is also labeled. So this can be quite important when you're trying to knock down a protein. So there you wouldn't want to knock down a protein in another cell type or in another part. So for example, there's muscle which is surrounding the gut, right? That's helping in peristalsis. So since this is just a red fluorescent protein and it doesn't interfere with any function, we can afford to express in all the organs. But when we are trying to knock down or make a mutation, we will express it in only the cells in the gut, and that we can do because there are drivers which are very specific to the gut. So you can use the proteins which express only in gut, use its promoters and express other things only in the gut. So you can do that. So that's a good question. Yeah.
Okay, sir. Thank you.
Joseph.
Yeah.
Yeah. Actually, out of the curiosity, I'm just asking one question, right? Okay. In 2019 to 21, even in the midst of the COVID times, there were many research papers related to the growing effect of gut microbiomes on Drosophila because of its particular diet.
Right.
So have you made any studies related to that?
Me personally, I don't look at host pathogen interactions, but the Drosophila gut is a very good model system for that, because you can imagine, you can just feed the fly with different pathogens, or you can infect it and then ask how the cells interact with it. But I imagine this live imaging setup will be very useful to study these interactions. So you could have a fluorescently labeled bacteria or something else, and then ask how the microbiome changes, how it interacts with the cells, and it could even modify how the stem cells are being divided and things like that. So I personally don't, but there are a lot of labs which do that. But I assume my system would be useful for them as well. Yeah.
Yeah, sure. Because there are many research outcomes yet just coming over, which directly reveals an interesting pattern that the Drosophila's diet is the best diet in this world. Because of the presence of a very unique microbiome or the microbial flora on its gut, which leads to the enhancement on the growth of the gut area too. So when you just specified that over here, I just asked. Okay.
Right. Thanks.
Thank you.
So, Joseph, I have another question. You said that you express the GFP only in the precursor cells, in the stem cells, right?
Yeah.
In the in vivo condition. How do you
Yes.
do that? Express specifically in the stem cells.
Right. Let me see if I have a slide. Let me just share my screen again.
Yeah.
Can you see this screen? So this is called an escargot flip out system. So what we are doing here is, how do I say? So there is this GFP. So this system is called GAL4 US system. So the GAL4 will bind to US and cause expression of whatever is downstream. For example, here it's the GFP. This is not the best diagram, but and this is the driver. So escargots are expressed only in the precursor cells, in intestinal stem cells and the enteroblast. And this GAL4 is under that. So this will bind to the US and then express GFP. But what we do is slightly more trickier, where you don't want it to always express, right? Because then over time, all the enterocytes will be labeled GFP. But you want to start the GFP when you want it. So what we do is there is GAL80, which is temperature sensitive, that can bind to GAL4 and keep it in a repressed state. So always it is in this repressed state. But like I said, it is in a temperature, it's a temperature sensitive thing. So you can now shift it to 29 degrees. So this fly is kept in 18 degree, and you shift this fly to 29 degree. What will happen is that this repressor will degrade off. So then, it is not binding the GAL4 anymore. So then the GAL4 is free to bind to this US. So then it will express the GFP there. So that is how we express it only in the stem cells or the precursor cells by just changing the temperature. So it's a very, so this is what you can do with flies, such intricate genetics, which I'm not an expert of because I just started working on Drosophila, but this is, I mean, you can do all sorts of things. So yeah. I don't know if I'm clear. If you have a doubt, you can just ask now. I mean, this is a bit complicated thing. It's not.
Okay, so it's right. Yeah.
So we have another question in the chat box from Umbly Elizabeth.
Right, I can check that. Why is this experiment done on live Drosophila? Is there any particular aspect which can only be studied on live ones? What about cell culture? Right. So what we want to see here is something what happens in a tissue, right? Here it's not one cell type. It is actually one cell becoming into another cell. And we want to understand how it happens in an animal. What happens in a cell culture is like extremely artificial in some sense. It's a glorified test tube, if you will. Because you can't control the composition there, the protein. I mean, all these are responding to a lot of different things. Even the stiffness of your glass that you keep in will modify what kind of genes are expressed in the cells. So we would prefer to do it in an animal. And in an animal, if you have to do it in a mouse or another organism, it's way more trickier. The Drosophila is easy that way. So that's why we are using Drosophila. And why live? Live is because you want to see its activity. How does one cell decide to become another cell, and what are the factors that regulates it, and how does this so you can see that it's like a triangular shaped cell, and then it becomes into these cuboidal cells with huge microvilli. So these things you need to study in a tissue. So this is a challenge, and that's why we want to develop ways to image it like how you can do with cell culture. Yeah. Thanks. Right.
Yeah, there's another question from Anjana.
Right. Why are we reversing the orientation after the expansion process? Yes, that's a very good question. So, okay, maybe I should just keep my slide on. Let's go to that slide so that I can explain this slightly more better. So usually, so what we are interested in this apical side, right? So, and usually we are imaging from the basal side of the cell. So this is how in microscopy, you have a slide and you have your cell like this. So your objective is going to be underneath here. Can you see my arrow? I don't know.
Yes.
Can you see this pointer? Yeah. So usually your objective is here, right? And you are imaging from below up. So, and this resolution is very bad when you image through your tissue. So this is what I was showing here. So if you image in Z, so this is XY, right? So imagine, it extends that way. So this is X, that is Y, and this is Z, right? So when you image in Z, your resolution is very bad in microscopy. Any microscope that you can take, even the most fanciest microscope, in this direction, it cannot image very well. So what you want to do, so imagine you cut your gut like this, right? And then you flip it down. So now instead of imaging above like that, this part will come down. So you're flipping it 90 degrees down. So this part will actually be down. I don't know if you are able to visualize this. So that's why we are cutting it and flipping it. And this cutting you can't really do if it's not expanded, right? Because it's so small. The gut is very small, and you cannot do there's no blade which is so small to cut it that way. So, but once it's expanded, we are able to cut it and then flip it. Yeah. So that is why. I don't know if I made it clear, but yes.
Yes, sir, it's clear. Thank you.
Right. Thanks.
We have another question from Sebin Jolly.
Yes, another question from Sebin. Is there any specific cell receptors, markers, which only intestinal stem cells display, which can be used to tag them? Yes, exactly. So, so as I said here, here we are using this marker to just mark the enteroblast and the stem cell, right? These this thing that I was showing here. So that's how we are just tagging the intestinal stem cells, and this is called escargot. So escargot is a stem cell regulator. It is expressed in stem cells. There is also another one called delta. These are not signaling pathway proteins, so you can use them to actually just tag only the stem cells. So you can use that and then just express it only in stem cells. But what we are interested in really is this one forming into this, so that's why we just label this using escargot. That's a very good question. Thank you.
So if you're done with the questions, Josie, we can go ahead.
Thank you very much, Dr. Joseph. That was a really interesting and enticing session. Every time we see such things, we know how complicated, how complex biological life is around us. And it creates more and more excitement for us to find new ways to decipher it. Thank you so much. To wind up this session, I invite Sharon Verghese, second year MSc Zoology, for the vote of thanks.
Thank you, Josie. Good evening, one and all. Respected Principal, Father Reggie P. Kurian, Head of the Department of Zoology, Dr. Jomohan KB, our Chief Guest, the Research Associate from the University of Cambridge, Dr. Joseph Thottacherry, the Organizing Committee, and all other participants. It is my honor to express my heartfelt gratitude to all the dignitaries and participants gathered here. To begin with, I would like to thank our Principal, Dr. Reggie P. Kurian, for blessing us with his presence and giving us immense support and encouragement for conducting this webinar as part of the centennial celebrations of St. Berchmans College, Thottacherry. The highlight of this webinar was no doubt the new ideas and knowledge imparted to us by our chief guest, Dr. Joseph Thottacherry. I extend my gratitude to you, sir, for enlightening us about the cutting-edge research tools regarding the study of epithelial stem cells. It sure was an interesting and comprehensive lecture, explained in a simple manner and useful to all students and scholars who aspire to conduct research in the field of cell and developmental biology. Thank you, sir, for spending your valuable time with us in the midst of all other commitments. Next, I thank Head of the Department of Zoology, Dr. Jomohan KB, for leading us in all our activities and being a great source of support and guidance. Special thanks to the Organizing Secretary, Dr. Philip Little Thomas, and all other members of the Organizing Committee for their hard work and efforts to make this webinar a fruitful event. Thank you to Josie J. Vincent from First MSc Zoology for the wonderful and organized comparing. No event is successful without a cooperative and attentive audience. I thank all the participants, including teachers, students, research scholars, for their attention and patience throughout. Once again, thanking each and every one. Thank you and have a great evening.
Yeah, thank you all. Thank you very much.
Right. Thank you very much for coming, and I hope next time we can meet in person when I'm in Thottacherry. And
yeah, sure. Yes.
Okay, thank you, sir.